because you want to get off anyway. So thank you very much for coming to give your seminar today. And everyone, please put your questions in the chat, and I'll monitor those at the end, and we'll put them onto Simon then. So thank you very much. Take it away, Simon.
Thank you very much. Okay, so thank you everyone for joining me today. I'm coming out of the sunshine to stare at your computer screen for an hour. I just wanted to talk to you today about the work we were doing in my lab for the last, I guess now, approaching 10 years, I suppose. I've been kind of running my own lab. And the interest I've got in particular type of gene control. And in essence, this type of gene control, post-transcriptional gene control, is all about balance in many ways, hence my choice of pictures for the start here. So I just want to, at the beginning, just kind of open up with the kind of concepts of this post-transcriptional gene control, just in kind of a basic manner, hopefully. So we must all, I assume, be familiar with the dogma of the gene to the protein process in biology, where we have our genes encoded by DNA and they contain the information for making proteins. And an intermediate to that, as I'm probably sure you're all aware, is the messenger RNA. So often when we're talking about gene expression, we can be talking about, often what we're measuring is the levels of the messenger RNA in a cell or a tissue, for instance. And we tend to typically measure the static levels. We look at a frozen point in time, looking at the levels of that messenger RNA for a particular gene, maybe. But the levels of messenger RNA in a cell are not really they're not being produced in a static process. So they're being produced by a process called transcription, where the DNA is read and the RNA is produced. But that isn't the end of the story. That they're also being turned over in a process you might want to call RNA turnover or RNA decay. And that's also a process that actually has a certain degree of regulation. It's quite strictly controlled by the cell. So the steady state levels that we'll mostly end up seeing in a cell of a given messenger RNA for a given gene is kind of analogous to this picture on the right of a bucket being filled with water, but a bucket that's got holes in. So the water coming into the bucket is the transcription of RNA being produced, but at the same time the cell is actively turning over the RNA using different mechanisms, and those mechanisms are represented by the kind of number and size of the holes in the bucket. And these two parameters together will end up giving us the overall level of RNA we might have in the cell at a given time. Now, this seems to be a kind of energetically poor approach, really. Why would you just make RNA only to just immediately destroy it, for instance, an hour or two later? It doesn't seem to make sense energetically. But it does actually make sense for another reason. So it makes sense when you think about the kinetics of the RNA and how you want to be able to turn genes on and off. So this has been known in theory for many years. So it's been shown that mRNAs theoretically with short half-life will be induced and turned off more quickly. Whereas mRNAs that have longer half-lives, that are more stable, will be induced more slowly, but their new level will remain much more stable once it's reached. And 10 years ago, David Baltimore's lab showed this in an experiment with using 3T3 fibroblasts constantly exposed to TNF-alpha. And despite a constant application of TNF-alpha, they found that there were different responses of genes when they did a microarray analysis of these cells. They had genes that were turned on and then off again very rapidly, genes that were turned on very slowly but steadily, and then intermediate genes as well. And they found when they looked at the half-lives of these genes, actually the genes that turned off and on very rapidly, such as the genes in group 1 here, these genes all generally had very short half-lives. Whereas the genes that turned on and remained high, these genes all had long half-lives. So the post-transcriptional control of an RNA, it's just a way that the cell means the cell can use to fine-tune its ability to respond to its environment above and beyond just a simple transcriptional input coming from the DNA to the RNA. And it's a process that is kind of very elegantly controlled at a number of levels by a number of factors. So just for as I work on cartilage, so I have to show a slide like this at the beginning of a talk. I'm not going to dwell on it because we've all seen slides like this before, but just to say that I'm interested in tissues of the synovial joint, and I'm aware there are many tissues of the synovial joint. Today is going to be pretty cartilage-centric, so a bit of bone thrown in. Cartilage is a deceptively simple-looking tissue. It is basically a group of cells in a rounded shape called chondrocytes, and they're embedded in a extracellular matrix that's aneural and avascular. And those cells do a pretty good job maintaining that tissue for many decades as a kind of shock absorber at the end of bones, and also it provides a smooth surface for articulation. But as I'm sure you're probably all aware, as we age, we're particularly susceptible to get diseases such as osteoarthritis, where this tissue breaks down. And the chondrocytes are singularly hopeless at dealing with this process. They don't really produce much of a repair tissue. Any they do produce just gets lost to the surrounding environment in the joint. And if they try and proliferate, they tend to proliferate all in one place, like a big gang together, rather than migrating to points of areas of damage. So understanding the process that goes on here is important, and also understanding what's happening in healthy tissue is important, because we want to really maintain this homeostasis early in life before this process occurs. So just an overview of the talk today. I just want to talk about very briefly first a sort of history of how I got interested into this type of gene regulation in articular cartilage or chondrocytes. Then I just want to talk about how this regulation is controlled in many ways in a disease such as osteoarthritis. Then I'm going to look at a modulator of post-transcriptional regulation control and how that controls development and function of musculoskeletal tissues. And then finally, if I have time, I'll be checking my watch. We'll also move on to looking at how another project that looks at inflammatory stimuli that regulate, particularly translational regulation, so how RNA translates into protein. We'll see whether we get there. So firstly, the history lesson. Many years ago in the 2000s, I'd say a lot of this work was done around 2004. I worked as a postdoc in Manchester in Tim Hardingham's lab in the UK Centre for Tissue Engineering, which funnily enough was linked with Liverpool. Manchester and Liverpool held that centre together. And my project was mainly looking at regulating a transcription factor called SOX9 in chondrocytes. SOX9 regulates a number of important chondrocyte genes. That's all you really need to know. It's important for the chondrocyte phenotype. And we were looking to see whether we could take chondrocytes out of their nice environment where they're embedded in this matrix and around, culture them in monolayer culture where they tend to lose their phenotype. They lose markers such as collagen 2 and they lose markers such as SOX9 itself. Could we promote SOX9 expression to boost other markers? We were using viral vectors to do this of various sorts. But as part of the project, I was wondering how we could just make the endogenous SOX9 be regulated rather than using a virus, because obviously we're looking at a tissue, problems that aren't fatal, problems of the joints that aren't necessarily fatal, and introducing gene therapy into those joints is going to be regulatory a big challenge for some time to come. So how could we regulate endogenous SOX9? And I was just doing some very simple, some would say quite basic experiments, looking at how I could modulate cell shape to regulate SOX9, because we knew that during the first few passages of monolayer culture, you could stimulate the chondrocyte phenotype again if you put the cells into a three-dimensional culture, such as this alginate bead culture. And the cells in this alginate bead is a bit blurry, but you can see we stained with alcium blue, and around these rounded cells, now the cells have got their round shape back, they're producing this blue matrix, which is staining glycoproteins in the cartilage matrix. And it's been shown by other groups that SOX9 is re-expressed in this process and is an important part of this process. So I was wondering how SOX9 might be regulated, and I did this really basic experiment, flawed in many ways, but it was just, I was looking and just trying to do various silly things to see how SOX9 might be regulated. And I wondered, well, heck, I can look at regulation of SOX9 over 48 hours in an alginate bead culture from monolayer, and I can see an increase in SOX9 expression. So I just asked, does that require new protein synthesis to occur? Is it intrinsic to the cell as it is? So I did a quite blunt thing, which I basically added cyclohexamide, which inhibits protein synthesis for 48 hours. And I mean, chondrocytes are pretty hardy beasts, and they survive that in general, when many cells wouldn't. But I really expected two things to happen. One, that nothing would happen at all, we'd still get the same induction, or two, that there'd be an inhibition of the induction. But actually what happened was the opposite, and actually got a much greater induction of SOX9. And that result there started me on this whole pathway of post-transcriptional regulation. Because from there, we basically determined that this process creates this induction of SOX9 by cyclohexamide as part of a broader process of stress-activated superinduction of SOX9. And we could see that with cyclohexamide and also with other stress stimuli such as hyperosmolarity, which is more of a relevant stimuli within the cartilage field. And what's actually happening here is that the decay of the RNA of SOX9 is not occurring as quickly. So these experiments here are called actinisin D chase experiments. We're adding an inhibitor of transcription and we're then looking at different time points after that. So because there's no new transcription, all we're seeing is the decay of the existing RNA. And you can see with both cyclohexamide and with our hyperosmotic stimuli of 550 milliosmoles, we get stabilization of the SOX9 mRNA. It has a longer half-life in those conditions. Without going into too much detail, we showed that MAP kinase signaling was important in this process, and particularly P38 MAP kinase, which this is an inhibitor of, significantly impaired this stabilization of the RNA. And then moving forward rapidly again, because I'm basically going through many years' work very quickly in one slide. Once I'd started in Liverpool and I had BSRC New Investigator Award, Ben McDermott came and worked with me under then. He was looking at how RNA binding proteins could maybe control SOX9 mRNA. It's to look for the molecular mechanisms that could control this stabilization of the RNA. And so, for instance, we found that a protein called TTP, or tristetraproline, was able to bind using these mobility shift assays. You can see these shifts here, particular sites within the RNA sequence of SOX9. And if we did a siRNA knockdown, this is a bit confusing, but I want you to look at just the circles here, which are the control cells where you get this nice decay of SOX9 RNA, and the open triangles here are where we've knocked down this TTP protein. So knocking down the TTP protein inhibits, stabilizes the RNA. So TTP seems to act as a negative regulator, causing the RNA to be turned over more quickly. So that in a nutshell is how we got. I wanted to give you a brief background of why we were interested in this whole process to begin with. So let's just move on quickly to how this gene regulation process might be controlled in a disease such as osteoarthritis. So I moved to Liverpool from Manchester. It's not worth going through the whole story, but it's partly involved with working with meeting Pete Clegg in Manchester when he was doing a research leave fellowship there, and then getting Arthritis Research UK funding with him that brought me to Liverpool first as a postdoc. And then we got a second set of funding, which led to this this piece of work, which was specifically aimed at looking at measuring RNA decay rates across the transcriptome in osteoarthritis. So we were taking tissue samples from normal or OA patients, and the OA samples we were also stratifying into OA into tissue that looked grossly normal when we received it, even though it came from an OA joint, or tissue that was fibrillated within the OA joint. I should also point out the normals here weren't age matched. These were from lower limb amputations due to bone tumors, and these were all much younger. So it wasn't a perfect match, but we take what we can when it comes to normal cartilage, human cartilage. So the basis of the experiment here was we isolated the chondrocytes overnight in collagenase, plated them out, and then basically used them 24 hours later. So basically using the minimum amount of time before we could use them. So we tried to fend off that dedifferentiation process as much as possible. And then we stimulated the cells using this actin-myosin D chase process, took RNA samples at different points, and then analyzed the RNA using microarrays. This was a long time ago, before RNA sequencing was being used. And so we were hoping to see how genes will either be stable transcripts, so over time we'd have a kind of very little change, or rapid change to causing unstable transcripts. And we could get two pieces of information actually from this experiment. Firstly, we could look at our 0-hour time points. This would be the same everyone else is doing, just looking at a time point with no stimulation of any sort. What's the effect of osteoarthritis, for instance, on just gene expression? Because that gives us the total mRNA level. But then the slope of the regression of this exponential regression here, we can use that then to provide it to calculate the mRNA half-life as well from this experiment. So this is quite different to what other people were doing, because we're actually looking at the kind of kinetic processes that are underway within the cell. And it makes it more challenging, because obviously you can't just get samples that are already frozen or dead. You have to get live samples, because the cells have to be alive while you're doing the analysis. So just looking at the overall mRNA levels, we were encouraged to see that we saw differences between both the intact and fibrillated OA compared to the normals. So we can see the coloured ones here, dots here, are significant changes in transcript expression. One of the interesting things is that when we took cells from either intact or badly fibrillated OA tissue, but they were both from OA joints, once they'd adapted to cell culture for 24 hours, they're almost identical transcriptome-wise. There's very, very little difference, which was quite an interesting observation. So look at the altered mRNA decay. increase the power a bit, we lumped the OAs actually together eventually in the intact norm, particularly because their transcriptomes are almost the same. We lumped these two data sets together to give a bit more power. And what you're seeing here is basically showing the frequency of RNAs with a particular half-life. So the half-life's along here, and the number of times we see RNAs with that half-life is up here in normal and total OA samples. You'll see on the right here is a bar here, which goes up really high. This is the analysis was capped at 24 hours. So any RNA that had its half-life calculated over 24 hours, which is put in the 24-hour bracket. So that line shoots up out of my top of my screen. So most of the RNAs are very stable, but there are unstable RNAs. And what the take-home message was we found that if this is just a zoom in the inset here, we saw an increased number of short-lived RNAs in the osteoarthritic samples than we did in the normal samples. So something was clearly going on there, changing the post-transcriptional control within these samples. We picked out some of our favorite RNAs from the lists that were regulated and checked them with qPCR and found, so here's just some examples of some of those RNAs. So we're particularly interested in groups of RNAs from the ADAMTS family, which are agrokinases, which are enzymes which are known to break down cartilage matrix. They were all destabilized in OA, so their transcripts were turned over more rapidly in OA. That might mean they're more susceptible to up and down regulation more rapidly in OA. Similarly, for transcripts such as hyaluronic acid synthases, there's a heparin sulfate sulfotransferase here, and a member of the NF-kappa B signaling family. So all quite interesting. And I want to link to there in a kind of quite slightly ham-fisted way to some work we've done more recently now, where we focused a bit more on ADAMTS5, because ADAMTS5 is one of the darlings of osteoarthritis research because it's been shown to be an absolutely critical proteinase, particularly in mice, for osteoarthritis to progress. And if you don't have activity of this enzyme, you don't get osteoarthritis in mice. So one thing I noticed over the intervening time after we did this work was there was some studies coming out that were telling a lot more about a process called alternative polyadenylation. Now, very quickly to explain this, and I don't want to get too detailed, every mRNA has a poly-A tail on its 3' end. So the RNA on the left-hand side, as we look at it, it's 5', to the right-hand side, as we look at it, it's 3'. At the 3' end, it has a poly-A tail. There's a load of A's tagged on the end. And this is very important for its stability. And generally, there's kind of one overarching site where this normally occurs. But for some mRNAs, this can occur in multiple sites. And this can lead to a situation where you have multiple copies of an RNA that code for the same protein, but actually their 3' untranslated region, this bit here that isn't translated, can interact with lots of different factors that are really important for controlling post-transcriptional control, factors such as microRNAs and RNA binding proteins. And you might get a gene where you have a subset of the RNAs that are actually immune to these regulators because they don't actually have this sequence because the poly-A tail's been added further 5', further up closer to the coding sequence. So there are various databases springing up showing from transcriptome analysis how this process might be occurring in various genes, and ADAMTS5 was a gene that seemed to be, from some of these databases, seemed to have some sites where this was occurring. So I had an Mrna's project with Connor Sugden, who many of you will know, who's now doing a PhD with Kevin Hamill. So he did a 10-week Mrna's project with me and really started some of this work off. So he was looking where we saw some of these sites might be. This very blurry image is terrible. This is just a terrible cartoon of what the end of the coding sequence of the 3' UTR of ADAMTS5's mRNA is. So this untranslated region doesn't code for protein, but possibly has many regulatory regions in it. And this blurry thing here is from the genome browser, just showing you how well conserved this region is between species. So you can see it's very well conserved after the coding region, very well conserved after it's what I'd call canonical poly-A site, but it has this conserved region here as well. So the databases were showing there might be other poly-A sites here, here, and here. And so Connor basically made assays with plasmid DNA that contained these sequences here, and then PCR assays that could look at expression levels of either side of these potential sites. And he was able to produce really nice standard curves with these assays. But ultimately in this project, I mean, we only had 10 weeks as an MWEF project. We couldn't really show any difference between either side of these, potentially where we thought these sites would be. And it wasn't until we continued working a little bit more with some undergraduate students the following year that it became clear that actually, if there was a site at all, it was actually around in between these two areas here and these two areas here, in this area here, around about, possibly around about 2,500 bases downstream from the stop codon. Whereas if we looked at expression levels along here, we saw this drop off, and we can see this quite consistently. And this happens the same in OA or normal. Green is OA samples, red are normal samples. There's no difference in OA here. We carried on and looked at some actinitin D chase experiments and found that this shorter version is actually more stable, which fits in with our idea that a longer version would have more sequence that could be interfered, could interact with regulators of post-transcriptional gene regulation. So what we're finding is that ADAMTS5, certainly in chondrocyte cells and culture, is existing with a kind of heterogeneous RNA pool that varies, and the two sets of RNAs, at least two sets of RNAs, vary in their half-lives. So Phaedra Winstanley-Zarek is a SEMA PhD student in the lab now, and she, during her MRes before she started her PhD, and slightly into her PhD to finish some of this off, she was looking at whether there were any factors that could control the ratio of this short and long version. As part of her project, she actually created some slightly more refined, just for various reasons, technically we thought we just needed to slightly change the assay slightly. And she's got, so 3 different positions here. She's measuring, actually she's only looking at 2 positions here. I think she's looking at 5 and mid in this experiment. And she was looking, in this case, looking at the ratio between the levels picked up by sequences here and the levels of sequences here. And she sees about a ratio of about 2 again, like we saw in the, so basically a 2-fold ratio between the short form and the long form. Which actually equates to, it's kind of complicated, because when we look at the short form, we're also looking at the long form, because of course the primers pick up both forms here. So when it's a 2-to-1 ratio, actually what we're looking at is more of a 1-to-1 ratio between a short form and a long form. Hopefully that's clear. It's probably a little confusing. But ultimately, what I want to take home from this is Phaedra did a lot of work, looked at a lot of samples, and we don't see any significant difference when we treat the cells, for instance, with interleukin-1, one of the cartilage biologists' favourite things to stimulate their cells with. Another favourite thing to stimulate our cells is with hypoxic conditions. Chondrocytes exist in hypoxic conditions in the joint. So when we put the cells back into hypoxic conditions, there does seem to be an increase potentially in the copy number of the ADAMTS5 in the hypoxic conditions. But if we normalise, this is just the same data again, but normalised slightly differently. So we're normalising to the 5' for each, either normoxia or hypoxia. We're basically normalising to this point here. We can see the drop-off in expression is the same for both normoxia and hypoxia. So the use of the site, the difference in expression, difference in the short and the long form isn't changed by those conditions. So without wanting Phaedra to bang ahead against a brick wall, constantly looking around at various different conditions that might or might not affect ADAMTS5, I was keen that we kind of changed and went for a slightly different approach. So we've kind of flipped the approach. So what we're looking at, and we're starting to look at actually, is how one effect, osteoarthritis, affects polyadenylation generally across the transcriptome. And the reason we can do that is because over the last couple of years there have been technological advances in RNA-seq technology, in particular this technology called quant-seq reverse, which I'm not going to go into in enormous detail because I'll probably mess up the process of describing it, plus we haven't got the time to go into it. But in effect what this does is it's an RNA sequencing process that only sequences, normally in RNA sequencing you'd sequence across the whole transcript from the 5' end to the 3' end. This only sequences tag regions at the end of the 3' end of the transcript. And it basically does that by initially reverse priming your RNA sample with an oligo DT that's an adapted oligo DT. You make a second strand cDNA library from this using random hexamers. But in essence, what you then do is you sequence backwards from this direction through the poly-A site across the join between the poly-A site and the sequence of the 3' untranslated region. I hope that's clear. What that tells you then is exactly the point in the RNA where the poly-A site is added to the transcript. So initially we had some help from Ibec, who's a PhD student in Mandy's group, and I'm one of his co-supervisors, and he's already got a really good method for extracting small amounts of RNA from small valuable cartilage samples. And so he already had some normal cartilage samples that we had obtained, and these actually this time they're age matched to potential OA samples. And we obtained osteoarthritic cartilage from a number of from our clinical collaborators at Clatterbridge Hospital in the Wirral. And we took 6 of those that were age matched to the normals and did the same process with those. Sent the RNA off to Lexogen, the company that runs this analysis. We've run these samples, we've actually sequenced them twice and combined the data to get deeper sequencing. So then the data analysis with those is done in 2 ways. It's the reads are aligned with star aligner, and then the differential expression generally is measured with DSeq2. So that's just to look at differential expression. So we can still use this to look at for where genes are upregulated or downregulated. But we can also use this analysis to look at the exact sites at which polyadenylation is occurring in different transcripts across the whole transcriptome. And with this, we're being assisted by a chap called Gregor Rott at the University of Zurich, who's developed this online tool called Express RNA. So this really is work in progress. We did our second sequencing of these samples during lockdown. Lexogen were open during lockdown. We did them again. So we're really at a very early stage of this at the moment. And so just please bear that in mind. And the follow-up, the data we're getting is going to give Phaedra enormous amounts to do as time goes on. So when we just look at the gene regulation, we're encouraged to see that we see lots of genes being upregulated, lots of genes being downregulated. Confusingly in this graph, because my R skills aren't good enough to flip the graph around, is that the ones at the bottom are upregulated in OA and the ones at the top are downregulated in OA. But we see lots of genes that we expect to be regulated. We also see a lot of novel transcripts regulated, and a lot of these turn out to be long non-coding RNAs, or potential long non-coding RNAs, that haven't been identified as being regulated in osteoarthritis before. And I don't know if it's a particular way, because of the way we're doing the sequencing, I don't know that they're coming out, but Phaedra's now designing some, now she's back in the lab, she's designed some assays to try and look at these with qPCR. But the really interesting part is the alternative polyadenylation patterns. And this is kind of a confusing graph, and this is really kind of raw data at the moment. So that one of the take-home messages from what we found at the moment, but Gregor, the collaborators, isn't particularly happy with the way it's been done at the moment, is that we're seeing quite a modest amount of regulation, really, as far as alternative polyadenylation between normal and OA. Now, Gregor thinks there's a lot of optimization that he can do with the alignment of the reads. It needs to be done specifically to be able to allow this to work properly. And he's kind of on the case with that at the moment. But from the initial alignments we've been doing that are probably not perfect, we do see some, so these coloured dots here are ones that his analysis identifies as being significantly regulated. But as you can see, it's not a lot at the moment. The output from this data, and these aren't ones that significantly regulated these genes, but the output comes like this, kind of confusing to look at, but basically you're getting a gene name. We get some quite interesting information. Most interesting, which is this is the site position on the chromosome where the alternative polyadenylation occurs. So that's the sequence point. So for instance, SOCS4 is on chromosome 14. It's on the sense strand, code on the sense strand. And so that base on the sense strand, if you went to a genome record, that's the base at which the polyadenylation site is in its 3' UTR. And it only has one polyadenylation site detected. However, a gene like ILF3 has 3 polyadenylation sites detected in this particular example. 2 in this 3' untranslated region, and 1 is an intronic polyadenylation site. And again, you get the exact number base point. So you can really get a really great idea of exactly where these polyadenylations are occurring. So just take very quickly, two examples, I think we included both, of ones that look like they might be regulated in OA. One is this KMT2A gene, which is a lysine methyltransferase, histone methyltransferase. So it's important in epigenetic regulation in the cell. It's actually downregulated in OA in our analysis. And if you look here, basically it's detected by the analysis in 2 forms, with a distal and a proximal polyadenylation site. So the RNA encodes the same protein, but its 3' UTR is longer or shorter. And you can see here in the controls, it expresses both proximal and distal sites. The distal site's slightly more than the proximal. There's quite a lot of this proximal version. But in the OA, the proximal version is almost completely lost. You only get this longer version of the gene, the mRNA being produced. And the implications for that, if you look at a microRNA prediction binding analysis and target scan, this is a bit of a mess, but this is the 3' UTR. Basically, what this loss is causing is a loss of all this regulation. Everything here is not being able to bind to the KMT2A RNA in this case. So basically what that means is in OA, all of the RNA is susceptible to these microRNAs, whereas a subset of the RNA in normal controls is not susceptible to these microRNAs, which is potentially very interesting. Just another quick example for ones that look like they're regulated is SMAD5, which is a Tgf-Beta signaling protein. This one's a bit more subtle, but in effect, it's kind of slightly opposite. Again, there's two forms with a proximal and distal polyadenylation site, and in this instance, the proximal control, the proximal version isn't expressed at all in normal samples, but it starts to be expressed in OA samples. But in this instance, does that really matter? Because actually, what we're losing in this proximal version is an area where there's not a great deal of microRNA binding, for instance. But there may be RNA binding, protein binding, or this microRNA 135 binding site might be very important for it. So this data is still very early, but it's uncovering some quite interesting concepts within in these OA samples. So in the interest of time, I'm not going to go into too much detail at the summary, but just to show that we see increased turnover of genes in osteoarthritic chondrocytes, and that we're also seeing changes in polyadenylation patterns, although they might be more limited, but might be quite important for particular genes, those patterns in osteoarthritic chondrocytes. So now I just want to quickly move on to the next section, which is looking at how modulators of post-transcriptional regulation can control development and function of musculoskeletal tissues. I should just say modulator. We're looking at one modulator. So that's an RNA binding protein called HUR. Its gene name is actually ILAVL1. So this protein has been shown to be essential for various stages of embryonic development by this paper coming out of Dimitris Kontoyanis' group. What caught the eye of this protein was one of the phenotypes that you get when you don't have proper expression of the protein in the developing mouse is that you get a skeletal phenotype. So if you make a germline knockout, you get no embryos beyond about 10 to 12.5, and a lot of them get resorbed by 12.5, embryonic day 12.5. And they have a problem producing properly produced placentas. So what Dimitris' group did, it's produce a conditional knockout where they knocked out specifically only in cells in the epiblast of the embryo. So these are the cells that are going to go on to form the embryo itself. And among a number of phenotypes, they saw the skeletal phenotype, which leads to problems with the development, the growth of limbs, for instance, and also patterning problems, since there's some syndactyly, they saw a fusion of the digits. And this sort of correlates or chimed with some of our slightly more modest work, where we'd, for instance, we had a student project, Connor Halloran, who'd been a vet student doing a summer project with us when we were still based at Leahurst, and he'd been looking at bone marrow stromal cells, human bone marrow stromal cells, and he'd been knocking out RNA binding proteins in those cells using siRNA, and he found in an osteogenesis assay. I used the word osteogenesis in commas because in case Rob or Jim are watching, because they'll probably dispute osteogenesis, but a matrix mineralization assay should be probably say. But in the assay, normally you get this red color when you stain the matrix, which shows it's mineralized. But when we knocked out HUR, that was inhibited, so it seemed to be affecting mineralization of the matrix. And then also in some of Ben's work, when he was looking at regulation of some genes by RNA binding proteins, he'd found quite consistently that MMP13, which is a known regulator of terminal differentiation of chondrocytes and that process during bone development, was upregulated when you knocked HUR down. So from these and a few other bits of data and the links with Dimitris, we wrote to Dimitris and he sent us his floxed mouse, which was the mouse model he was using in the previous paper. For those who don't know about flox mice, I'm not going to go into massive detail, but what it basically means is the flox means that these modifications either side of the gene, and they don't do anything normally. But if you cross the mouse with a mouse that expresses a particular enzyme called CRE, which is normally found in mice, the CRE can cut these two sites and basically lop out this bit in the middle. So you basically knock the gene out. And then you can get CRE mice that express this enzyme that expresses enzyme specifically in different tissues. So what you can effectively do is knock the gene out in a specific tissue, depending on which CRE mice you use. So we went forth and did that. So one of the CRE mice we used to cross with this HUR floxed mouse was a PRX1 CRE. And this is quite a common CRE used in the skeletal development field. It was developed by a guy called Malcolm Logan, who's a Brit, who's at King's College, but he worked in Cliff Tabin's lab in Harvard, where a lot of kind of top developmental skeletal biology came out in the 90s and ongoing. And they showed that PRX1-CRE was expressed in the mesenchymal tissues within the developing limb bud. So these lumps here are the developing limb bud. And the black marking is where the stain of the PRX1-CRE is, and it's expressed in these regions. So we went and crossed the mice because that sounded like a good, much more specific than knocking out in the epiblast. And the first couple of crosses we did, we started finding mice with quite a strong phenotype, but they were really small, and we couldn't understand why they were so small. They actually looked much more like the kind of mice that Demetrius was seeing in his previous paper with the SOX2-CRE. And we couldn't understand why the whole mouse was small. It shouldn't just be affecting the limb buds and maybe the craniofacial region. And they had highly disrupted limbs. The development was really poor. And quite embarrassingly, I'd failed to read the minutiae of the paper from Malcolm's paper, because if I'd paid a bit more attention, I'd have realized that in this figure here was showing that if you derive the CRE during the breeding from the mother, you can get a sporadic germline delivery of the CRE. And that's what we think is happening here. We think, because it turned out that the two breedings we did just happened to be, we hadn't paid attention, we just put mice together we thought conveniently put together. And you know, you can tell my naivety as a mouse biologist here in this first project I've managed in this way, in that we'd use mothers to provide the CRE. And actually, to get this kind of effect reproducibly, you needed to use paternal CRE delivery. So we went forward and did that. Kirsty Johnson was doing this as part of her PhD, and then Craig Keenan also came on board for a couple of months and was helping with some of this work. But the take-home message, when we delivered the CRE paternally, so this now should be knocking out this HUR protein in just the developing limb bud mesenchymal tissue. And this is an embryo here that's got the right genotype for that to happen, and there was no difference to its littermates. It looked exactly the same. When we did histology on the limbs, they look the same. There's no difference between a CRE positive animal and a CRE negative animal. When Craig did immunohistochemistry, we found that indeed HUR protein was lost. And I don't know how well you can see, you might be able to see it here, slightly better here. In this region, this cartilaginous region here, it's very few cells stained compared to the control, where it's ubiquitous. HUR is all over the place, and it's really noticeably knocked out and then pretty much by E16.5 in most of the mesenchymal derived tissues, it's not present anymore. So the CRE was working, but it just was showing that HUR actually isn't necessary for development of the limb, certainly from the point it's knocked out about E9.5, E10.5. At the same time, Ben McDermott was doing a study looking at knockdown of HUR in primary murine chondrocytes, basically taking cells from these mice, from xiphoid to these mice, and then knocking the HUR out by delivering CRE by an adenovirus. And by doing that, he could take the adenoviral CRE treated cells, then had HUR that was easily about 80–90% reduced compared to the controls. And when we did RNA sequencing on that, in line with the previous result in vivo, we actually found very little change in the gene expression in chondrocytes, primary chondrocytes that have been treated in this way. So those that had less HUR, there were a few upregulated and a few downregulated genes, but nowhere near as many as we thought there would be. So this was kind of a bit of a surprise. It wasn't what we were really expecting. It kind of took the wind out of the sails slightly, this. At the same time, Kirsty in her PhD was looking at a different CRE model, which is one of George's CRE models, which was the CRE is driven by an aggregam promoter, so it's expressed in chondrocytes. But it's inducible, so you need to provide tamoxifen into the mice to make it knock the gene out. So basically you can grow the mice to adulthood, for instance, and then knock it out later on. And that's what Kirsty did. She grew these, she grew the mice to adulthood, injected them with tamoxifen, and then I think this would have been 5 weeks later. She looked at, this might be 2 weeks later, sorry, I'll have to check that now actually. But what she saw was recombination a few weeks later. At the genomic level, she saw a combination caused by the CRE. She saw loss of HUR and articular cartilage by Western blotting, and also a certain degree of loss of HUR. And immunos are terrible here because our sections kept falling off the slides, but compared to the controls where we see HUR is expressed in the cartilage, we still see it expressed in the cartilage, but there's lots of non-expressing cells present as well. So we're getting certainly a partial knockdown of HUR induced in this system. If we look at 5 weeks, that doesn't actually affect the cartilage and the joints at all. This isn't no challenge, just looking after the knockdown. So then we did an OA model, a DMM model of OA, which is the mechanical model of OA. And we found that in both the wild types and the HUR mice, we saw a degeneration. And if you can see here, this degeneration of the cartilage tissue as compared to the controls, sham controls, but there was no significant difference between the knockout mice and the wild type mice. So again, kind of disappointing. So we weren't really seeing any effect of, certainly as far as the cartilage level, any effect of HUR expression. Another experiment Kirsty done was to look at development and how this, in using George's cray model, how this could affect knocking HUR out using this could affect development of the mice. So she injected pregnant mother at E9.5 for tamoxifen and then collected the embryos at E16.5. She could detect in the embryos, for instance here, an embryo had cray expression in its ribs and its tail, and then she looked at using our recombination assay and she saw a recombination in the rib but not the tail. So hopefully these videos work. So what we saw then was that actually we did see some differences, but it was only a very sporadic difference. So these micro-CTs hopefully show it. These are from 2 different litters. And sporadically we saw this examples of embryos like this that are Cre positive. And compared to the wild types, the mineralization of their ribs and spine was severely impaired. But this wasn't the same for all, this doesn't happen for all Cre positive mice. So there's clearly a huge degree of variability going on here. But this was the first inkling that actually there was some kind of thing happening. HR was in some way controlling the skeletal system. So more recently, I've had an undergraduate student last year, and I just wanted to talk about this work because Hannah did an absolutely sterling job as an undergraduate student in the last semester, just before the lockdown. And her project was really to look at a different tissue structure in the limb bud, not the mesenchyme, but at the ectoderm that's at the edge of the mesenchyme, because this region, a region called the apical ectodermal ridge, and this, if you imagine a limb bud growing out this way, this region of ectoderm is actually critical for the signaling processes that control both the proximal distal and anterior posterior patterning of the limb. So we know that FGF8 is a marker of this. So she wanted to see if FGF8 signaling was changed in these HUR knockout mice. So she did such a great job because she not only did she do the whole process from beginning to end with help from Kerr and George's lab to isolate the embryos and some help from David Turner with the in situ hybridization technique, and the protocol for it. She pretty much did the whole thing herself, made her own probes from scratch, and got some really beautiful images of 10.5 embryos, and particularly these ones here. These are wild types. Seeing the crescent-shaped appearance of the staining for this FGF8 at the ectoderm at the very edge of the limb bud. So we looked, this time we were using PRX1 cray again, and we used the mothers purposefully to try and get a complete knockout and get these very small embryos. Annoyingly, the embryos weren't this time. They weren't that much smaller at 10.5, and it was really unclear whether we were actually getting this time a full kind of germline knockout, or whether this kind of sporadic nature of this meant this time we were getting mesenchyme knockout like we do with the paternal delivery of cray. Certainly in this, you had these 2 embryos that Hannah looked at here, and one embryo, the AER, the apical ectoderm, which was perfectly fine, absolutely normal. This embryo, maybe it's a little bit patchy. It's hard to make any firm conclusions from this. The other angle that I was taking with this, and really at the end of the MRC grant that I had that ended last year, I kind of bet all my money on red and spent the rest of the money I had on it getting this mouse, this MSX2 cray mouse, which is supposed to express the cray recombinase in the apical ectodermal ridge. And I wanted to see, is this region a key region that HR is important in for controlling limb development? So we bred this, got this mouse in at great expense from Charles River, bred this mouse into the Phlox line. But at this F1 point here, we're basically breeding our Phlox mouse into the Cre mouse. The Cre mouse doesn't have the Phlox sites. So because this is Phlox homozygote, all of our F1 progeny have to be heterozygote. They have to be heterozygote. They'll get at least one of these alleles from the parent. And to cut a long story short, Transnetics' genotyping for the BSU showed that they were all wild type. They all came out as wild type. I even took culled a load of the animals. And this was whether they were cray positive or negative as well. I took a load of these animals and did the analysis myself. I'm not going to describe it, but that basically what that shows is they were all wild type. They were all coming out as wild type. This was just before lockdown here. I'd spent my technology directorate voucher pretty much in getting to this stage. It's a huge amount of woe, to tell you the truth. transgenetics are actually very helpful, and they used our recombination assay. We told them what it was, and they used it, and they also did some sequencing of the samples. They basically found that the animals were all heterozygous for a recombined flox allele, even if they were Cray negative, which was really annoying. So I don't know what kind of Charles River have sold me here, but whatever it is, I'm breeding into the flox allele, and it's just automatically knocking out the flox allele, regardless of whether there's Cray there or not. Cue my anger. So we're doing this repeat of this breeding one more time, and those mice are just being weaned. We'll genotype them, and depending on whether it's good or bad news, or depending on whether we continue with this or knock it on the head. So the final point of good news in this story is, like, just before knockdown, we had these mice that were PRX Cree, paternally derived, so they were normal. They looked normal when they developed, and so they were born and they appeared normal, and they grew up and they appeared normal. And basically, we culled them at 6 months, and we were going to just do histology on them and look at the cartilage and bone, etc. But I did micro CT on them just because the opportunity there is there to do it. And so this is a wild type littermate, and the Cray negatives. This is normal micro CT. And then when we looked at the micro CT of the Cray positive animals, there was a really striking difference between the wild types and these Cray positive animals. We had 2 males, and there's a female here that is more severely affected. So the trabecular bone in these animals is really, really disrupted. The cortical bone seems to be normal. A better bone biologist than me would be able to tell me whether that's different or not. But certainly trabecular bone, even to the naked eye, to a non-expert, is very, very different. And so this is an area we really need to look at now, and I need to work out how I'm going to get the resources to be able to do this working forward. So time-wise, because I started 3 minutes late, I might try and squeeze the next section in within 5 minutes if I can, if it doesn't annoy people too much. So I might just skip over the summary of that so we don't waste too much time, and hopefully you recall what I said there. And then just move over to how the inflammatory stimuli regulate, particularly translational gene regulation. So in joints, inflammatory stimulation is a thing. In osteoarthritis, it wasn't always thought to be an important part of the disease, but more and more it's now understood to be a factor involved in osteoarthritis. And for many years, cytokines such as interleukin-1 have been used as model systems for inducing degradation of cartilage, so that we can learn more about how that process occurs. But those model systems use very high levels of IL-1, and they use them for quite extended periods of time. What's more, clinical treatments targeting interleukin-1 have been shown that they don't affect osteoarthritis outcomes. Also, animal models with interleukin-1 signaling that's been impaired actually aren't protected against osteoarthritis. In many cases, often they get worse osteoarthritis when you apply out osteoarthritis models to them, which is a bit of a conundrum. With that as a background, in addition, we had some data going into this that seemed to show that very early responses to IL-1 could upregulate anabolic factors such as SOX9. So over maybe 1 to 3 hours, we see upregulation of these factors rather than the downregulation that is what people more normally expect of the effect of IL-1. So with the background of that, we were, a few years ago, Brian McDonough and I were discussing doing this process of riboseq analysis on some samples. And so we decided to do this on IL-1 treated chondrocytes, or chondrocytic cells, I should say, because the cell type we chose to use was this chondrosarcoma cell line, which isn't that much like a chondrocyte, but it does actually replicate IL-1 responsiveness quite well to the responsiveness of chondrocytes. In essence, what this process does is it takes, you basically take the RNA that's inside the ribosomes at a given time within the cell. You basically freeze the ribosomes on the RNA. So these are the processes, these are the things that are making the proteins. So the RNA is inside these structures. And then you digest away the RNA. The RNA inside the structures is protected from that digestion, and then you isolate that RNA and then you sequence it. And what you're looking at then is the RNA that's being actively translated in the cell, as opposed to the RNA that's just the total RNA in the cell. We used a kit from Illumina. This isn't a box of the kit. This isn't a picture of the kit because you can't get a picture of the kit anymore because they discontinued it because it was such a load of rubbish. And it was a heroic effort by Ben to work with the people at Illumina and the tech supports there and make up his own approaches that he managed to get this whole process working, to say the truth. And then we did data analysis via the RiboGalaxy platform. I'm not in the interest, I might show this as just a kind of thing you do when you do ribo profile analysis to show it's worked okay. In a nutshell, what we found was that the these are now we're looking at genes that are translationally regulated. So the RNA might not be significantly regulated in its overall level, but it's being translated into protein a lot more or less. And genes that were particularly affected by that, and we just did this string analysis just to kind of group them together, was groups of genes that were involved with actual ribosome assembly and function, and groups of genes that were more, particularly interestingly, groups of interesting genes that were inflammatory associated. And we've done proteomic analysis of parallel cultures. We found there was a number of chemokines and cytokines that were both translationally regulated, but also secreted at higher levels into the media following IL-1 stimulation. This just shows that articular chondrocytes, primary chondrocytes, also have the same stimulation and secretion of these factors. I won't go into the details of that actually in the interest of time. So this moves me on really towards nearly the end now, because I just want to talk briefly about work that Becky Horn and the lab's been doing. So she's another PhD student, BBSLC DTP funded student. She's kind of taken some of this work on now. She's repeated some of the work that Ben had been doing with ELISA work, looking at secretion of these translationally regulated cytokines in these cell types. And what she's in essence finding is this kind of dynamic range of concentrations of IL-1 where you go from no response as far as a secretion of a factor like IL-6 is concerned to maximal response. That dynamic range is actually not the 10 nanograms per mil that most people would use in their experiments, but it's actually from 1 picogram to 100 picograms per mil. And it's much more in line with the ranges you might expect to see from normal into damaged or injured joints. Even though you get quite a lot of expression, say at 10 picograms per mil, and it's in many cases bordering on the levels you see you kind of saturate at. If you look at, for instance, say some signaling pathways, such as MAP kinase signaling pathways, you see quite a big difference. You get a very rapid increase in signaling, as many people know with IL-1. Within 10 minutes, at high concentration of IL-1, you get a rapid increase in signaling of, say, this phospho P38 MAP kinase or this phospho junk is much more strongly seen. But the induction of these pathways in the lower levels, at the lower concentrations, even though you're getting quite a lot of IL-6 secreted still, is actually quite low. Obviously, we've just looked at particular pathways. There's lots of pathways that can be regulated. And what we're hoping to do is maybe do a phosphoproteomic analysis of this and look at the low and high concentrations as Becky's PhD moves forward. Finally, it's on to my final slide now. It's quite an interesting experiment that Becky did where she was looking at how IL-1 stimulation over short periods of time affects an overall response over a 24-hour period. So what she's done here is she's looked at 24-hour periods of exposure, or a 24-hour culture period, but the IL-1 has only been present for either 1, 3, 6, or for the full 24 hours. And she's measured the release of IL-6 at each of these points where it's been removed, or she's measured it after 24 hours. And you can see that by 3 hours, after only just 3 hours in pretty much most of these replicates, what we're looking at here is low concentration in this column, 10 picograms per mil or 10 nanograms per mil in this column. These are just 3 experimental replicates, same experiment. And what you generally see is that by 3 hours, you have what is going to be, even if you take the IL-1 away at this point, after 24 hours, you pretty much have, in most cases, bordering on the maximal amount of gene expression over 24 hours. Sometimes it requires 6 hours. But what it seems to show is that there's a period of time, a reasonably short period of time, and then beyond that, further stimulation with the cytokine isn't required, and the cell in many ways retains a sort of secretory memory for at least 24 hours beyond that time point, which potentially has implications in how we understand how fluctuating levels of inflammatory cytokines might affect cells, of chondrocytes particularly, in the joint. So I just wanted to finish there, just to summarize that part, and that we have interleukin-1 beta is clearly altering translational rates very rapidly. So there are clearly things happening in response to interleukin-1 beta in chondrocytes that control translational rates very quickly. And a lot of these things are things like other inflammatory factors, that there might be a cascade of processes that are being regulated very quickly in this manner. We're looking into the signaling that's occurring there. There's very different levels of signaling depending on the concentration you look at. And then there's evidence also that this whole process only requires very short periods of time where IL-1 is exposed, and low levels of IL-1, not the high levels for longer periods of time. They're typically used in a lot of the literature. So I'll just finish there. thank the people, particularly the people in bold in the lab who've done a lot of the work involved in this. And then some of the collaborators, Demetrius for saying the mice that we used, Gregor for his ongoing work on the alternative polyadenylation data, David and Daryl assist with Becky and Phaedra's PhDs respectively, Brian for assisting with the work together on the IL-1 work we just talked about, and also Julie Aspton, who we're just about to start working with on a new Dunhill funded grant looking at translation and polyadenylation and how RNA decay rates are affected by age specifically rather than by a disease associated with age. And I'll just thank these funders, and I can finish there. Thank you very much.
Thank you, Simon. Very rapid translation through a whole lot of stuff. There are a couple of questions, and I'm going to get people to ask them themselves. So, Richard, you want to ask your question? I'm not sure whether you got your answer or not.
I think. Thank you, Simon. I thought that was really interesting, and it got me thinking about a lot of things. So, one of the questions I had really was earlier on. You were talking about the stability of the different messenger RNAs, and you particularly mentioned poly-A, and then you talked about length a little bit also. Do you have a feeling or a rule of thumb that we could use that would estimate the stability of mRNA from something that was measurable from the sequence, i.e. its length and its actual sequence?
I don't think it would be possible to necessarily predict exactly how the decay or the translation rate of the RNA would be affected based simply upon whether it had sequences or didn't have sequence. We could hypothesize and infer that, but I think we'd have to experimentally measure whether that was actually happening at this stage. What we could do is take those, it's hard to do, as I said earlier on, it's hard to do the kinetic analysis of the RNA decay that we could use to directly compare with that data. What I'd like to do is is taking this data forward once we've finalized a bit to try and go back to our original RNA decay work in OA and see if there are any areas that can resolve together, and whether we can see patterns where we see changes in RNA decay that marry up with alterations in polyadenylation. But I'm going to wait. I'm really trying to wait off at the moment until Gregor especially has weaved his magic on this data.
Okay, thanks.
Mandy, do you want to ask your question?
Yeah, so Simon, using your quant-seq reverse data, just thinking about your differentially expressed mRNAs, how different do they look compared to other studies where they generally look at young normal versus old OA? So the changes that you see are likely to be OA and not OA and aging at the same time. Yeah,
that's a good question. So I haven't done that for all the normal genes yet. What I have done is that there was a paper actually you shared with me, Andy, looking at long non-coding RNAs and how they're regulated in OA. And as I recall, that paper came out a couple of years ago, those samples were not age matched, whereas ours are. And I compared all the long non-coding RNAs and they showed were regulated in OA with the ones that we're showing now are regulated. And I think of the 15 or so they had, the 3 of them we only picked up 3 of them as being regulated that they'd shown. So there's clearly quite a bit of difference. And I think that's kind of strength actually of this data set. And it wasn't actually, I hadn't quite realized actually that the lack of this comparison between age-matched samples exists. And so I think that's actually strength of this data set above and beyond the polyadenylation analysis is actually the ability to compare these 2 age-matched cohorts, which hasn't actually been done before using this kind of technology.
No, so that's exactly what I was thinking, because they are age matched in most studies. I mean, I don't think there are any of my age matched studies. People don't do it because they can't get hold of normal old tissue. Yeah,
and then, because Ibex had so much practice, in his approach to getting the tissue and getting the RNA from the tissue, it was a really good time to be doing it, I think, because, we've got Ibex around and he's got that skill at the moment, which we need to give someone else as well when Ibex goes.
Thanks, Simon.
So, Simon, can I ask you a question it's just from an ignorant point of view, really? So when you've got a mouse like this and you've modified it and you're looking at a joint that's got lots of different tissues in it, and then you look at how the modifications affect one tissue or another tissue, how do you try and understand how the effect on one tissue might actually be changing the effect on another tissue?
That's a good question, really. I think what we tend to do is we tend to take the primary way of analysing the outcomes of these models is to use histology and to use some quite well-established scoring systems that have been laid down over the last 10 to 15 years in the field, so that everyone is pretty much using the same scoring approach when they come to analyse these samples. And those scoring approaches take into account not only changes to the cartilage, degradation of the cartilage, but also changes to bone, to the formation of osteophytes on the edges of the joint, and also to synovial structure as well. And we didn't see any difference in any of these factors in the analysis we did.
Okay. So you can kind of separate them out then?
We do sort of separate them out, and they I guess they're all doing their own thing, and they're all, in many ways, they're all communicating with each other as well, I suppose. And in many ways, that's one of the great unknowns, I suppose, of osteoarthritis, is how these things are all managing and interacting with each other.
Okay. Anyone else? No more questions? Well, thank you very much, Simon. I guess actually we've got to past 2 o'clock, so I expect people have got to go to other meetings. Yes. And you need to go as well. So thank you very much. And I think it's Malcolm giving his talk next week. So I hope to see you all again then. Thank you a lot.
Thank you very much.